Hi, everybody. Thanks for coming back. We're starting a couple minutes late. It's my job. I'm Cristen Willer, University of Michigan. It's my job to introduce the next session. We'll be hearing from three outstanding scientists talking to us about analytic methods. I'm supposed to remind you that we're going straight through till 4, so hopefully you all went potty before you got here. First speaker, Nicole Soranzo from the Wellcome Sanger Institute.
It's really a great pleasure and a great meeting. Happy to talk here. So I was asked to talk a little bit about data analytics, and so I'm not really going to talk about my own work, but rather try to give you a perspective of the field, what we've been doing in the last 10 to 15 years, and potentially some of the challenges to come. Before I start, I just wanted to make a very obvious, perhaps, but a really important point, is that methods don't exist in isolation, and indeed are intimately linked to data growth. So if you take any given biological problem, you have a new hypothesis, you generate some pilot data, develop a new method, there's a technology scale up, a large-scale project, you learn more about the problem, you devise new hypotheses, and so on and so forth. And so this is far from a linear process. It is indeed potentially a very nicely expanding problem. And so as a community, we really need to think about what are the technologies or the analytical that really can provide step changes either in the acceleration of this process of discovery or indeed in acceleration of learning about these problems. And really, this is my feeling is that, the idea is that the ICDA will provide really the ideal platform for data scientists to come together with functional genomicists and different type of biologists to think together about what are the ways in which we can leverage analytical methods together with new data to boost this progress. And I think really we need to think hard about among all the possibility of data sets we can generate and indeed methods we can devise, what are the ones that are more likely to be really game changers? And again, thinking about this in a slightly out-of-the-box fashion. So fundamentally, here we're trying to address 4 main questions. The first important question is what are the genetic variants that affect disease risk and patient outcomes, defined quite broadly. Through which mechanism, ideally causal mechanism, do these variants act? Do these variants have invariable effects, or do they have effects that manifest only in given conditions on environment? And ultimately, why we're here is what are the analytical solutions that will allow us to accelerate understanding of disease risk and patient outcomes. And I just want to go through some of these questions in a little bit more detail. So with respect to the first question, this is really a question of discovery. And I really would like to give a shout out to the humble GWAS, because this is really the reason why we're here in this room. And it's been really quite remarkable, the scope of discoveries we've done using relatively simple statistical frameworks such as linear regression or logistic regression. But indeed, obviously, there are many fairly sophisticated algorithms that went behind collecting some of this information that we use, for instance, in data calling for genotypes or phasing and imputation. And so what we learned is that indeed GWASs are relatively easy when it comes to disease predisposition and risk factors, but indeed still very difficult for studying patient outcomes, including disease course, severity, response to treatment. And here we really need to think about how we can enhance our ability to capture relevant phenotypes for a discovery. So the other big theme over the last few years was the association of rare variants, and this was really prompted by the fact we discovered that GWAS don't account for all the heritability of traits, and the advent of genome sequencing allowed us finally to look into the space of low frequency, high effect, rare variants. And obviously these are very difficult to study because they are rare, often rare or private or population specific. They have a known allelic architecture, and also the interpretation of the functional consequences, particularly outside genomes, is really difficult. And this makes it really difficult to model association tests. And indeed, because of these challenges, there have been really a huge amount of effort put into developing ever more sophisticated statistical tests that have different founding principles and explore different underlying allelic architecture. And despite the fact that we've done a little bit better with this test, they have not been a game changer in terms of understanding or discovering rare variants. And this is clearly a problem of needing more data rather than better methods. And obviously, we are now at the stage where the genome sequencing are nearly quickly outpacing the increase of discovery of genome-wide association studies, and indeed, it's inevitable that this will lead to more rare variant discoveries. So now analysis of association has become really effectively a computational problem rather than so much of a genetic problem. And again, thanks to Vani Rivas, we can think about highly integrated, high throughput computational infrastructure to allow us to not only do association testing, but indeed look at downstream effects such as survival, relatedness, and logistic regression, and indeed to visualize and represent the data in the best possible way. There's perhaps something still to be learned about rare variants, and this is really coming from the fact that we're learning a lot about, from population gene sequencing studies, about some of the properties of the genome, and particularly looking at prediction of deleteriousness of different types of variants, what are the allelic spectra, where selective pressures act, and how do these selective pressure act to, for instance, flatten effect sizes for associations in different genes, depending indeed on their causal assumption on the trait. And so we can begin to use this information to define perhaps better genomic and biological priors that we can feed into methods such as particularly Bayesian methods that allow us to iteratively scan non-coding regions of the genome. So the second big question, and there was been a lot, sorry, this is really distracting. The second big question has been a lot of talk about this is through which causal mechanisms do these variants act. And we heard a lot about the power of phenotyping in cohorts. I guess the point I want to make here is that we learned particularly that there are many regions of the genome that control association with very many phenotypes, what we call pleiotropic regions. But really our understanding to take these findings forward has come with the development of better and better ways of estimating SNP heritability genome-wide or for select sets of variants in a way that is, for instance, robust to LD structure or other types of confounding. And so what this is telling us, what this is giving us is, first of all, a better ability to quantify what proportion of the heritability of traits we're currently explaining using our genetic variants, but also to really begin to understand biological correlation between traits and between traits and diseases. And this is just a plot of genetic correlation calculated using LD score regression across a number of phenotypes. And what this is telling us is allows us to take away the effect of stratification and confounding and allow us to evaluate what is the genetic sharing genome-wide between different phenotypes. It also allows us to get better genome-wide priors about what type of biological mechanisms are likely to underpin some of these associations. And for instance, we can see that often pleiotropic effects at trait-associated variants are underpinned by pleiotropic effects of regulatory information. So really, what we want to, beside the correlation, is really to nail down on causation. Again, methods to test causation have been around for quite a while. And here is perhaps the simplest formulation of how we go about looking for causation, Mendelian randomization framework, as proposed by George Davis Smith and colleagues many years ago. And what this is a model that has been shown to be robust under many assumptions. One of the critical assumptions that we now know is violated, particularly now that we have access to multiple layers of information, is that the assumption that an instrumental variable acts on an outcome through a modifiable exposure is often violated in the case of correlation pleiotropy. So particularly Stephen Burgess and colleagues have developed a number of diagnostic approaches that allow us to test the violation to the assumption and overcome some of these limitations. But it is clear that this is not a solved problem, and we need to be better at inferring causality. There are other more recent approaches that also look at this problem with slightly different ways, and this is an approach by Alkes, Price, and colleagues that uses a latent causal variable model to estimate effectively the percentage of causality between two phenotypes given a structure such as the one shown here. And so this is now allowing us not only to have a yes and no answer as to whether a trait is causal to another trait, but rather to estimate the level to which one trait might be contributing causally to another. What is great now is that there are now many computational platforms that allow us to look at Mendelian randomization and causality quite easily, and indeed the Mendelian randomization of everything against everything means that now we can access this information without learning how to do this properly, but also be with less risk of making a mistake while doing so. So the issue of causality is not only at the level of traits or genome-wide, but it's also the level of loci, and we heard a lot about the use of EQTLs and colocalization approaches to look at causality at the level of individual variants. And indeed, many colocalization approaches are based on Bayesian statistics, and this has been proven to be also extremely useful for fine mapping association variants. But these are still very painstaking efforts of taking one variant at a time and one trait at a time and colocalizing and looking at evidence. And it is clear that we need to do better at that. And we need to do better both by developing a multivariate fine mapping or colocalization methods, but also finding better representations of complex biological phenotypes. So this is a paper by Oliver Stegel and colleagues, which is using effectively a very nice and elegant extension to the basic principle component analysis to effectively take multiple layers of information from the same people and factorize them into this type of matrix, so that indeed the same type of data across layers can be represented by a matrix of factors by sample and a matrix of factor by feature. And this is nice because it allows us to address issues such as data sparsity and indeed the imputation of missing value, and also, for instance, to identify phenotypic outliers across large numbers of phenotypes. So these are really nice approaches, and it is clear that we will be more and more reliant in the future on them. So where we need to go, and this has also been spoken about a lot, is the point where at some point we'll be able to take a genetic map and a small representation data set, such as a gene expression in a given cell, and develop approaches that allow us to impute phenotypes and do a phenome-wide association study in cohorts and disease patients. And this has already been done by this approach and TIWA's approach and this extension on gene expression, but it would be really nice if we can combine information that I showed you before with the existing approaches to begin to impute across different types of data. All right, so the other point I wanted to make is that obviously, all of this is about a cross-sectional exploration in a given time point, but we really know that humans are complex, they are dynamic, they are environment responsive, and what we really barely scrape the surface of is how genetic variants affect expression phenotypes, expression of human phenotypes in different contexts. And so this is clearly important because this is a way in which we can address real biology and potentially get closer to phenotypes such as disease predisposition, disease progression, or drug treatment. So one way in which I think we're going to end up doing this is through polygenic risk scores, simply because, like a heritability measure, they are a handy way to look at sets of variants in a relatively simple way. And indeed, there's already evidence for polygenic risk scores that are indeed variant depending on, for instance, the age of the subject in this case, the sex, or different types of environmental exposures. And so really what we want to do is that we want to get better at integrating the functional genomic data, for instance, that are coming from EQTLs, with polygenic risk scores to define biologically risk scores that are informed by biology or different types. And this could be biology that is informed by traits measured in the resting state, but also biology that is informed by our growing understanding of physiological changes in non-resting states, in perturbed states. And I define here perturbed states as very generically as, for instance, a disease perturbation, a demographic perturbation, developmental stage, an environmental, or even a genomic perturbation. So really, we're getting to the point where we can begin to play with genomes and cellular system in a slightly more interchangeable ways compared to what we have done until now. So in vitro, we typically use resting cells that are isolated from donors, and we subject them to perturbation, for instance, in the case drug and a stimulus, and we might measure phenotypes, such as, for instance, gene expression phenotype or some functional phenotype of interest. We can think about using similar, and genomic perturbation. We can think about using similar approaches, in this case in vivo, and obviously the hope is that electronic health records and molecular phenotyping done longitudinally will at some point take us to a stage where we feel we are measuring environments well. But there are things we can do in the meanwhile, and one of the obvious one is to look at clinical trials where we have a very controlled perturbation with a before and after status. And obviously in this case, we don't want to look at noisy phenotypes such as drug pharmacokinetics, but rather molecular phenotypes that are perhaps bigger effect sizes and more responsive to the phenotype. So really, it'd be nice to get to a point where we can think about how we can leverage the power of data both to in vivo settings and in vitro settings, and potentially combining the two to get a better handle about the effect of the environment. The other thing that we need to do in order to do this is also to have better ways of looking at measuring environmental effects. And this is, again, a nice method by Oliver Stegel and Rachel Moore, who has effectively devised a statistical method to do genome by environment scans. But in the particular case, the environment is measured through simply a covariance matrix, which can be as discrete or as continuous as you want it to be. And it is indeed continuous to the extent to which the environment is continuous. And so this allows you to map QTL associations, in this case for the discrete case with only two types of effects, but potentially with an unlimited number of effects. So it's really nice that we can begin to think about more flexible statistical approaches to try and mimic some of the complexity of biological data. And there are many other challenges ahead, and we're thinking about now and perhaps the near future is to take into better account the disease heterogeneity. But projects such as the Human Cell Atlas are telling us that biology is even more complex than that. We know there are non-discrete biological states, which can be inferred through this type of equation from single-cell RNA-seq data, but there are also spatial relationships within cells that create microenvironments, and it is possible that also cells will respond to those environments differently. And I'm not for the moment arguing that we should jump into this for genetic association. These are very complex problems, but perhaps thinking about some of the complexity that we'll be facing in the future. So as I said, the other point is to see, if we want to really devise transformational approaches, this is not only about transformation analytics, but also about transformational data. And so one of the things that we need to think about is how can we re-engineer existing techniques or methodology for large-scale analysis to get closer to functional phenotypes. So as you know, I work on blood, or some of you know I work on blood. And so in blood, we are working with Sysmex analyzers, and we know that from Sysmex analyzers, you can get access to raw data that measure the angles of this diffraction of the light. in a flow cytometry setting. And it's been shown through clinical use that some of these parameters that you can measure are indeed related to, for instance, intrinsic properties of the cell, such as granularity and nucleic acid content. So can we re-engineer this automatic seismic analyzer to look at functional phenotypes? And the answer is yes, we've done this. And indeed, you see that if you compare the association you get from this type of data to traditional full block measurement, you see that for white cell component, many of the associations are new, are indeed associations that are related to functions such as, for instance, antibiotic defense. So are there other opportunities that we're missing to get one step closer to function in a high throughput, controlled, and quantitative way? So with this, I'd like to basically conclude by saying there are analytical solutions that will allow us to accelerate understanding of disease outcomes. The list of priority changes all the time. So these are, at the moment, I try to list some of the problems I think we're facing, problems we have a good handle on, problems that we have a good handle on where we're still missing a lot of data to address, problems we are beginning to face as the data becomes bigger, and indeed problems that are just beginning to understand. And this is why it's so important the analytics community joins very closely with the data generation one. So in terms of developing data commons, as well as analytical methods, we talked a lot about diverse population, where diversity is not only defined as ethnic diversity, but also as the age, environmental diversity of disease status, where genomic data sets are large, they are single cell resolved, they are interoperable, where we're looking at phenotypes not only at one time point, but along a longitudinal trajectory, and where we test the function ultimately of every variant in the genome in every cell. From the analytical point of view, there are a number of methods that have served us particularly well. These are some simple frequentist methods, but also linear mixed models, variance decomposition methods, Bayesian methods, and I think we'll learn later about deep learning. And really what we want to do is tailor these methods to do phenome-wide and genetic scans, to impute, infer, and integrate across unmeasured states, to assess causality, to look at interoperability of phenotypes between cohorts, and ultimately to predict the function of every variant in the genome in every cell. And with this, I'd like to thank you for your attention.
Thank you, Nicole. That was an absolutely wonderful overview. I feel like I need to watch it again in slow motion to absorb everything you covered there. Fantastic. So our next speaker is Anshul Kundaje from Stanford University.
Okay, so I just want to start by thanking the organizers for this opportunity. So Olga already gave a really nice introduction to some pioneering work that her lab has done using deep learning models trained on regulatory genomics data to sort of prioritize non-coding variation. My talk is going to focus on another aspect of these models. What are these models actually learning? So can we actually gain some interesting biological insight from these kinds of so-called black box models? So just to put things in context, as we all know, large-scale regulatory genomic profiling of hundreds of different biochemical markers across the genome in hundreds of cell types have produced comprehensive catalogs of regulatory elements and their context-specific activity. Clearly, in the talks that we saw over the last 2 days, these data sets have been very powerful for a variety of different applications. They also serve as fantastic data sets to train statistical and machine learning models. And I'm going to focus on two key applications. One is sort of to take these relatively low resolution annotations and make them base resolution. So really get extremely high resolution context specific sequence annotations. And the second is sort of try to also understand what they're doing, why they are potentially powerful, what are the advantages when they're being used to interpret regulatory genetic variation. So just to introduce the problem, the way the way predictive models of regulatory DNA are built are sort of complementary to the association testing approach. In the association testing approach, you typically have a variant, you look at its variation across many individuals, and you associate that with some molecular disease phenotype. And that's why you would require usually very large cohorts, because you have one position and many variation across many individuals. The way these models work is you take one assay, for example, let's say a ChIP-seq or a chromatin accessibility assay, and you get observations genome-wide. And your goal is to learn a predictive mapping from DNA sequence to these molecular profiles. And so the way you can think of this as a machine learning problem is you can bin the genome into little chunks and then take the sequences of those chunks as inputs to the model, and your goal is to map those sequence inputs to binary or continuous summaries of activity within those bins. So typically 100 base pair bins or 1,000 base pair bins or something of that sort is used. And that's, there's a very large number of models of this type, including the very nice ones that Olga presented in her talk. Now, one thing I'll argue is that we are actually not leveraging the full power of these assays. And the reason I say that is because if you really look at the fine scale profiles at base resolution, each of these assays like ChIP-seq, ChIP-exo, Nexus, DNA-seq, ATAC-seq, they produce these absolutely gorgeous distributions of reads, and the exact shape of these reads, what often people refer to as footprints and so forth, capture exquisite information about protein DNA contacts. And by summarizing information in large blocks of 100 base pairs and so forth, you just lost all that information. The other reason why this is important is because, for example, and this is not a criticism of ENCODE because I actually work very closely with ENCODE, but the classical approach for annotating regulatory elements is to take these assays called peaks, and then sort of you have this arbitrary delimitation of peaks. And here's an example, and this is very common actually in the genome, a particular enhancer or a region in the genome that has these DNase accessible sites in a variety of very different cell types and tissues. And these elements will often be called as one element. They're the same because they are overlapping. But the real question is, are these really the same? Is the DNA sequence regulating these elements the same way or not? And the answer for this element is no, they are not the same, even though they look like they're very similar. And you can get a sense for this by looking at the expansion of the profiles and the relative spacing of the summits and so forth. So all I'm saying is there is a lot of information in the shape of these profiles that is missed by existing models. So what we've done is recently, this is very fresh. We have a bioarchive paper that's just released last month. We built a new model called BPNet, which can actually map DNA sequence to regulatory profiles at single nucleotide resolution. So we start off with DNA sequence, something in the range of 1 kb to 100 kb, and we map this entire sequence to stranded or unstranded read counts directly at single nucleotide resolution. And the models, what they do is they take raw DNA sequence as the input, they learn features, sequence motif-like detectors, and each of these neurons learn these sequence motif-like detectors automatically by training on the data. And the higher layers in the model capture combinatorial interactions. So they can capture more complex sequence grammars directly from the data and map them to these base resolution profiles. So just to give an example of what this looks like, this is ChIP-exo data for 4 transcription factors, OX, OX, NANOG, and KLF in mouse CS cells. And the tracks you see on top, let me see if I can. So the tracks you see on top are the observed tracks, and what you see at the bottom are the ones that are predicted by the model. This is on sequences never seen by the model in training. And in most cases, it's hard to distinguish the observed from the predicted. So if you, and these are of course cherry-picked loci, but if you characterize the performance of the models genome-wide, at least in terms of predicting the shape of the profiles, they equal replicate accuracy. So if you compare two replicates, how similar they would be, the sequence model is as accurate as replicates at the level of predicting shapes. For total occupancy, they're not as great, but there's a good reason for that. I can discuss it offline. That's because, well, the short answer is local sequence is not enough to predict occupancy. You need more information, but the shape of the profiles can be predicted perfectly by the DNA sequence. So we can get really accurate base resolution models, and the question is, okay, so we have nice predictions. What are these models actually learning? So my student Avanti Srikumar, she spent the last 4 years building absolutely phenomenal methods for interpreting these so-called black box models. One of them is called DeepLift, published in 2017. The other is TF Modisco, which is actually unpublished but on preprints. And we and others have been using these methods for interpreting the models. So I'm not going to get into the details of how the methods work, but I'm going to show you examples of what they can do. So here's an example. Let's say we looked at this OCT4 distal enhancer in mouse ES cells, and we saw this really nice this element, and we see these gorgeous footprints of OCT4, of SOX2, of NANOG and KLF, and all 4 transcription factors bind the exact same enhancer right next to each other. So very difficult to distinguish what exactly is driving binding of each of these. If you take the models and you reverse engineer which nucleotides in the sequence are contributing to the profiles, predicted profiles of these 4 transcription factors, you will see that they can really nicely capture the OX-SOX heterodimer, NANOG, a novel NANOG motif right next door, and these KLF motifs pop up right there. So you can take any single enhancer in the genome in a specific cellular context and just de novo interpret the precise contributions of every nucleotide in that sequence. And you can do this for any enhancer in any cell type. Now what's cool is you can not just go to regular enhancers in the genome, actually we can do this also in repetitive elements. So we've looked at all transposable elements in mouse, and we see absolutely phenomenal combinatorial binding of just these 4 transcription factors, and it's way more complex if you look at more. You can see different pieces of the transposable element being used by different factors to bind, and the binding sites within these repetitive elements are actually quite different from the canonical ones you see because of the age of the repetitive elements and so forth. What you can also do is then summarize these motifs. We have a new motif discovery algorithm. We can summarize this across the whole genome, and we're looking at 4 transcription factors, and the typical approach would be 4 TFs mapped to 4 motifs. The answer is actually 4 TFs mapped to 51 motifs. And these are not just random motifs. They're supported by distinct footprints in the chip exodata. So combinatorial footprints in the data. There are several novel ones. So for NANOG, we find 3 different binding modes. And these binding modes are supported by crystal structure. So if you look at the way the protein binds the DNA, the precise positions that have the strong importance in the motif of these 3 motifs really are the ones in contact with the NANOG binding domain. And we have lots of other interesting novel insights in the paper if you want to take a look. What we also find is for NANOG, we find a novel 10.5 base pair periodic binding pattern. So here you can see, if you look at the central motif, this TCA piece, you see these really lovely plumes of AT-rich sequence, which are predicted by the model to be very important for NANOG binding. That is exactly 10.5 base pairs, which is the helical turn of DNA. And not just that, but NANOG actually also likes to bind as homodimers at a 10.5 base pair preferred spacing. So either 10.5 or multiples of 10.5. And you can see that right here. And it's not just a prediction of the model. If you take the model and then look at the read counts at the exact same frequency, you can also see it in the reads. But that's only if you center it appropriately across the genome at these predicted sites. So it's not just like the model's not just imagining this, but it's actually present in the read data. And a nice way to think about this is that the prediction that we have is that NANOG is binding as a homodimer on these nucleosomes at 10.5 or 10.9 base pair periodicity. We can also use the models to discover motif syntax. Now, so far I've talked about individual motifs, but we know that regulatory elements are driven by highly cooperative binding of transcription factors. And so what we can do is, since the model is so accurate at predicting nucleotide resolution footprints, we can actually perform in silico experiments. So here's an example of what I call an in silico MPRA. We create a synthetic sequence, and we place 2 motifs or 2 ends, and we start moving them towards each other. And the model starts predicting what would happen to the footprints of all the different TFs as the sequence changes. And if you map this out, this is what it looks like. So what you're seeing is that this is the influence of the OX-SOX motif on NANOG binding as you change the distance between the motifs up to 150 base pairs. And you once again see very strong effects, very periodic effects. That's again a 10.5 base pair helical periodicity. And this is unidirectional. So OX-SOX has a very strong effect on NANOG. But if you look at NANOG's effect on OX-SOX, there's no effect. It's a classic pioneer factor effect of the OX-SOX pioneer factor affecting NANOG directionally at a very 10.5 base pair preferred spacing. We can do this also on sequences in the genome. So this is what we call in silico CRISPR. So you take real genomic sequences. Here's an actual enhancer. You see an oct and nanog, oxox and nanog footprints and motifs. You can delete the oxox motif and see what happens. And you see nanog and oxox both disappear in terms of binding. You can delete nanog, and it has no influence on oxox. And if you again quantitate this across the whole genome, you again see the exact same 10.5 base pair pattern. So the in silico sort of synthetic sequence models actually agree with what you see in vivo, on the real genomic sequences. And finally, just to conclude on this piece, it's not just that we're predicting binding well. We can use the same models to predict reporter activity. So actually activity of reporter sequences that we've never seen before in the exact same cell line. And the binding models perform very well on expression prediction as well. So that's sort of an insight into how we can take these black box models and actually discover very interesting novel biology at very high resolution by using interpretation methods. I also want to give a little vignette on how these models can help you interpret what regulatory variants might be doing. So as you know, there have been a variety of QTL SNPs found in the genome in different cell types and tissues. Often the causal variant is hard to figure out, but if you, in this particular paper by Terranci et al. From Hunter Fraser's group, they identified binding QTLs. These are SNPs that directly affect ChIP-seq of these various transcription factors. And most of these SNPs are likely causal because they are within the binding site itself. However, they have this interesting statement in the paper that about 1 to 10% of these binding QTLs do not overlap known motifs. And so we wonder how this might be happening. What's the mechanism through which these variants might be actually affecting binding? So I'll give you a small case study and then actually with the global result. Here's an example of a QTL that affects binding of a protein called SPY1. The position of the SNP is right here, the G. And if you overlap this with sort of known motifs of SPY1, if you align them to the sequence, you see that they match most positions except at this position, which is a T. And the motif, the classical motif of SPY1 says that it cannot tolerate a T at this position at all. The way to think about this is if you computed the score of match of this instance of the genome to this motif, it would be an extremely low score. So often these kinds of positions where you have even single base pair mismatches at positions where that nucleotide cannot be tolerated according to the canonical motif, it appears like these SNPs are not lying within canonical motifs. But this is what the neural network says. It says that all of these bases are favored except the T, right? And actually what you see is 10% of the sites that overlap the BQTLs, in fact, do not have an A or a C or a G at that position, but rather have an A or a T. And this is a small example where there are a large number of positions in the genome where you have low affinity binding sites which don't match the canonical motifs. And canonical motif models are typically very poor at capturing these low affinity binding sites. And this is really the advantage of agnostic models like neural networks that learn from raw de novo sequence. They do not make these assumptions and they can learn a large proportion of these low affinity sites. And genome-wide, just to show you again, the PWMs and the deep learning models behave very similarly at high affinity sites, but really it's in the low affinity regime that the nets really show a significant advantage. So I'm just going to close by speaking a bit about how we can improve these models going forward. I think one of the key things is the models can do a lot of things very well. My personal view is that they're not really there yet in terms of being able to predict effects of non-coding variation, especially weak effect sizes, very accurately. And the reason partly is that they're training across the genome on effect sizes that are much larger than what you see in terms of variation at a single position across individuals. And so what we really need to push these models to the next level is to be able to train them on multiple molecular profiles, directly on explicit natural or engineered perturbations, such that this input to the model are these personal genomes or edited sequences. And so the model really gets to see the effects of editing of a sequence on molecular phenotypes. I think the other interesting aspect is the machine learning community hasn't been particularly integrating well with the statistical genetics community. And I think there's really interesting scope to integrate molecular DNNs like the ones I showed with statistical genetics models, such that these kinds of models can really go completely end-to-end. They don't just stop at the molecular layer, but they fit into statistical genetics models that can push them all the way to predict disease phenotype across the entire spectrum. I think one thing that Olga also mentioned that I'd like to reinforce is we should think of the genome locally as a sequence and globally as a graph. And so far, all the, even the machine learning, neural network models and so forth, typically use sequence models to model stuff across the genome. Each element is modeled independently of everything else. And there are very powerful neural network models called graph neural networks that allow us to model these sort of more long-range physical, functional, trans interactions across the genome. There's lots of exciting work that could be done in that space. A big problem that all machine learning models have in genomics is, and probably most predictive models in genomics, is that they're actually quite poor at predicting out of distribution. So an example is if you train something in one cell type, it doesn't generalize to another cell type. If you train something in one ancestry group, it doesn't generalize to another ancestry group. And there are, I think, powerful methods in machine learning, domain adaptation techniques that allow these models to generalize out of distribution. New interpretation methods will continue to provide novel insights into the cis-regulatory code and mechanisms of variant effects. And finally, I think as we use better approaches to account for the esoteric noise distributions of each of these molecular profiles and confounders, a lot of these predictive models will get better. And lastly, I just want to close with one small thing. I briefly mentioned yesterday about we've discussed a lot about repositories for data, and we have repositories for publications. As I said, hinted yesterday, there's actually really quite a need also for unified repositories for models, such that it's very easy to use these models to make hypotheses and then compare hypotheses in a single framework. And so we've built this, the first model zoo for genomics. We call it Kipi, which means garden in Greek, like a zoo. This was just published in Nature Biotech a few months ago, and it already hosts about 2,000 different models, and you can use these very easily with a few lines of code to predict variant effects, to predict transcription factor binding, a variety of different molecular phenotypes. And we're starting to include also models that can predict other higher order phenotypes from sequence into these kinds of repositories. So if you're producing these kinds of models, please feel free to get in touch with me and add your models to these zoos. So I'll just stop there and thank all my students who've done all this hard work and my funding sources. Thanks a lot.
That was another outstanding talk. So we're saving our questions till the end of this next speaker, and then we'll have a few minutes to discuss what you've heard. So next up is Hilary Finucane from the Broad Institute.
Thanks very much to the organizers for giving me this opportunity to present. In a genome-wide association study, at a given genomic locus, we typically imagine that there's a very small handful of variants that have a non-negligible true causal effect, and that because of linkage disequilibrium and noise, what we actually see in our association data is a much more complex and noisy pattern of association that's harder to interpret. Statistical fine mapping is the process of using statistical models to try to go back to this top picture, which is the one that we'd really like to be looking at as we try to do biology. And there's a lot of reasons that we want to do this. The first is sometimes you'll find a putatively causal coding variant directly implicating a gene. Sometimes you'll find a putatively causal regulatory variant that together with some kind of enhancer gene map or EQTL data will implicate a gene. But beyond just the gene name, fine mapping can help you identify a variant to gene mechanism that, for example, can allow for the design of more realistic experimental systems. And finally, by looking at patterns of putative causal variance across the genome, you can start to look for aggregate signal, and this can be particularly important for cross-population analyses because the differences in LD and minor allele frequency across different populations can lead to much more dissimilarity among association statistics than you think there actually is in the true causal variance. Unfortunately, fine mapping is a difficult undertaking. Even if you have enough samples to identify a genome-wide significant locus, you need increasingly more sample size to disentangle these closely correlated variants. So one thing that I think ICDA should keep in mind is we can't declare victory just because we found the region. We need to keep on increasing our sample size so that we're well powered to actually resolve the causal signal at the region. Jointly modeling multiple causal variants at a locus is a complex undertaking, both statistically and computationally. Fortunately, there's been a lot of work in this area, and there are some now some really good and sophisticated algorithms like FineMap and SUSY, which are the ones that I'll focus on in this talk. That they were developed in Maddie Pirinen and Matthew Stevens' groups, respectively. And the third reason that fine mapping is hard is that it's very sensitive to input. So, for example, if you need to input the LD pattern in the population that you're studying, but because we're trying to make these very fine-grained distinctions, often 1,000 genomes, for example, won't actually give you accurate results. Previous fine mapping studies have led to insights about a range of different phenotypes, including IBD, prostate cancer, hematopoiesis, and autoimmune disease. My group wanted to take advantage of these large-scale, very well-powered GWASs that we have for many phenotypes in the UK Biobank and in Biobank Japan, and we've been very excited to be collaborating with Biobank Japan on this project. Masahiro Kanai and Jacob Eulersch are the two folks who have been leading this. They're grad students in my group, co-advised respectively by Mark Daly and Pardis Sabeti. And they undertook to fine map 15,000 loci from these different phenotypes in these two different biobanks. For those who are interested in the technical details, they ran FineMap and SUSY, allowing up to 10 causal variants per locus using mixed linear model summary statistics, in-sample dosage LD, and very generous region definitions. Fine mapping algorithms report, among other things, a posterior inclusion probability, which quantifies the probability after we've seen all of the data that we actually think that the variant is causal. And the first question that we asked is, do these PIPs agree across the two different methods? And what we found reassuringly was yes, that the large majority of variants that we included were deemed unlikely to be causal by both methods together, and of the remaining variant trait pairs, only 4% differed by over 5%. And so we decided to restrict our subsequent analyses to variant trait pairs where the two methods agreed. Then one thing you can ask is, well, how much resolution did we really find? How many variants did we find that had high PIP? And so the median number of causal variants per locus in this analysis was 15,000. And so being able to say for a variant that it has a posterior probability of causality or a PIP over 10% is actually something that we find very exciting. And we found over 25,000 variant trait pairs that had PIP over 10% and approximately 3,500 that had a PIP over 90%. So these are rather confident findings. And then we asked, what are these variants? So we took the genome and divided it into five categories shown here, and the genome-wide average is displayed here on the left. And so then we could take all of the variants that we fine-mapped and bin them by PIP. So on the left-hand side are the ones that we think are least likely to be causal, and on the right-hand side are the ones that we think are most likely to be causal. And we made sure that each bin has at least 200 variants. And what you can see is that as we get increasingly confident that a variant is causal, we're seeing large enrichments for these functional categories, especially the exonic variants. We could then use this pattern of enrichment to ask when we, was our approach of restricting to results where these two methods agree, are we throwing away mostly noise, or are we throwing away mostly signal? And so here on the left is the plot that I just showed you, the variance that we're reporting analyses for, which shows this very nice pattern of enrichment. But then if you look at the variance that we're throwing out, where the methods disagree, neither method seems to be showing a very strong signal of enrichment. And you can kind of see this most easily if you look at the leftmost side and the rightmost side of this plot. So what we would like to see is that our most confident causal variants look different than our most confident non-causal variants. And that's true for our kind of high confidence these methods are agreeing, and not true for when the methods disagree as much. We found a lot of fine-mapped coding variants, and so these are the variants that directly implicate a gene for a particular trait, and we found tens of genes with coding variants for tens of phenotypes, led by height, which has 97 genes that are implicated by fine-mapped coding variants in this analysis. You can then flip this around and ask for each gene, how many phenotypes did we find to be associated? And we found a large number of highly pleiotropic genes. So 24 genes had coding variants for more than 10 different phenotypes. And this question of pleiotropy has come up several times, and here are some concrete examples. So I want to walk through one example of the kinds of things that we're finding in this analysis. So here's the ABCG2 locus for uric acid in UK Biobank, and ABCG2 is a known gene for a related phenotype. And then here's the same locus in Biobank Japan, and you can see that there's very strong signals of association, but it's not quite clear if it's the same causal variants or different causal variants. Following fine mapping, this is now the PIP on the y-axis, we found in UK Biobank 2 variants that were very high confidence as the causal variants driving the association. In Biobank Japan, there was a slightly more complex structure where the method's estimating that there's something like 4 causal variants with different levels of confidence. It turns out that the 2 variants from UK Biobank are both missense variants, and that the top 3 variants in Biobank Japan are also coding variants. And so we can now visualize them along the ABCG2 gene. And so here, each variant is colored by the phenotype that it's being reported as putatively causal, with the shape reflecting the population it was found in. And here, there's a third biobank, FinGen. So Masa has also been fine mapping FinGen in collaboration with the folks there. And so these results now reflect that collaboration as well. And now we can walk through and ask, okay, so what are all of these variants? On the left, you can see 2 missense variants, both of which are putatively causal in UK Biobank, not found in the other biobanks because they're rarer monomorphic. Moving to the right, we have a missense variant found in Biobank Japan that's rarer monomorphic in UK Biobank in Finland. There's another missense variant here that's common in all populations but wasn't confidently fine mapped in all 3 populations. And then there's a stop gain variant that's common only in Biobank Japan. And so you can see here that we have an allelic series of 5 different coding variants that we were only able to put together because we were analyzing all of these different biobanks together. And this is actually not a very unusual gene. There's over 30 genes that have an allelic series of at least 4 coding variants, and over 60 genes that have an allelic series of over 3 causal variants. And just to contrast ABCG2 with another gene, ApoE, this is what it looks like when you have 2 missense variants that determine your ApoE allele and that are associated with a large number of phenotypes in all 3 biobanks. So I now want to take a minute to discuss a very recent analysis that we just undertook. So in a recent edition of the American Journal of Human Genetics, Terry Manolio exhorted us to analyze not just the white British subset of the UK Biobank, but all of the UK Biobank. And we found this very compelling in my group, and so I asked Nathan Chang if he would be willing to take this on. And so he performed a GWAS of mean corpuscular volume, which is the phenotype that we saw the very best fine mapping results out of all of the phenotypes that we looked at in both of the biobanks. And so he looked at MCV as a phenotype in UK Biobank and added not just the rest of the European samples, but also large South Asian, East Asian, and African cohorts that exist in that biobank. So this is only about a 10% increase in sample size. And the first thing that we asked was, are we finding the same thing? If you just take the variance and you plot the significance level in the original white British analysis from the NeoLab against this new multi-ancestry analysis, then do you get the same thing? And for most variants, there's actually quite good agreement, but then there are some variants where all of a sudden we're seeing the significance shoot up. And so I asked Nathan, what are those variants? It turns out that they fall into only 2 loci in the genome. Moreover, those 2 loci are the alpha globin locus and the beta globin locus. And now if you look at the beta globin locus, the top 2 variants are a known beta thalassemia variant and the most common sickle cell allele. And so we see this as a really strong proof of principle that Terry Manolio was right, and that we actually will be able to get a lot out of analyzing all of the different populations in the UK Biobank, even though we're only increasing our sample size by about 10%. So we're very excited to be collaborating with some of the world experts on multi-ancestry analysis, Alicia Martin and Elizabeth Atkinson, and together with them, we're planning to compute multi-ancestry stats for many UK Biobank phenotypes and to release them publicly. Then we're going to compute matching LD, which is an important point I'll come back to in a second. And while I don't anticipate that we'll be able to release that publicly, we are planning to get in touch with the UK Biobank and ask if there's some way to at least make it easily accessible to folks who have an approved application. And then we'll use this LD and summary statistics to find map to do multi-ancestry fine mapping in the UK Biobank, and this is all in collaboration with the NeoLab. So I have 2 points that I want to make here for ICDA. One point is regards this question of what is the appropriate LD, and I think this is really important moving forward. So one thing that's been highlighted are some of the analyses that we're able to do with summary statistics and reference panel LD, and many folks in the methods development community have been embracing this paradigm. The problem is that that only works when you have good reference LD. And so I want to distinguish here between an imputation reference panel and a reference panel for GWAS-based methods, where what you're actually doing is approximating the in-sample correlation with correlation that you get from some reference panel. And moving forward, as we try to make fine-grained biological statements, as we try to analyze low-frequency variants, but especially as we try to start analyzing cohorts like the UK Biobank, not restricted to white British ancestry individuals, the idea that we're going to be able to use a reference panel is going to get increasingly challenging and, in my opinion, unrealistic. And so I think that one thing that I hope that this group as a whole thinks about is, as we're deciding what we're going to try to make it easy to share and what we're going to try to make sure folks have access to, that in-sample LD is high on the list. Because there's nothing more depressing than when someone comes to you as a statistical geneticist and says, I have this awesome, huge new GWAS, can I run your methods? And you say, yes, well, step 1 is, can you please remove all of the non-European ancestry samples, and then we can talk, because you don't have in-sample LD and there's nothing I can do with your data. So that's one thing that I really hope as a community that we keep in mind as we're looking forward. And the second thing is, my group is now about to embark on some co-localization analyses based on the fine mapping that we've done. And I don't think we're going to be able to do this very effectively for either Biobank Japan or the non-European fine mapped variants that we found from UK Biobank. And so I think, again, prioritizing which things we need in multiple populations and which things we don't, things that depend on natural genetic variation like QTL studies, is something where we are really going to have trouble moving beyond the initial discovery if we don't have these complementary QTL data sets in the other populations. So with that, I'd like to thank Masa, Jacob, and Nathan, who led the analyses that I presented here, as well as our collaborators at Biobank Japan, FinGen, and my funding sources. Thanks.
So we'll invite the 3 speakers to come up for our discussion question. Please approach the microphone if you want to be first. So just to remind you, we have Nicole Soranzo, Anshul Kundaje, and Hilary Finucane. Yes.
Hilary, great talk. Have you looked at the effect sizes comparing different populations when you have the same confident defined map variant in multiple different populations?
Yes, and it agrees quite well.
Yeah, we've seen the same thing in EQTL data, which is good.
Yes.
So those were excellent talks. Slightly to both Hilary and Anshul. have you got a sense of what will happen, imagining kind of incredibly high European populations? So there's one question about being multiple populations, but can you do any kind of discovery statistics on how many how many base pairs we can absolutely functionally implicate as being the causal base pairs as we increase sample size? So I was wondering whether you thought about doing this in a staged way of increasing the sample size and seeing how your discovery curve would have worked out. There's sort of an equivalent question to Anshul, but now not using genetic data, but using this kind of epigenomics data. So both of them are trying to get at how much data do we need to get to before we saturate in these different samples.
So I'm happy to answer the first half of that. So it depends on the LD, and you can actually, there's analytical expressions, so you can write it down. You don't even have to simulate, and I forget what exactly they are, but for any fixed LD pattern, then eventually you should be able to do it very well. The way I see it is the more your sample size increases, the more fine-grained the distinctions you'll be able to do. So it's more like some loci become now tractable that previously had LD that was too tractable. And I think for any sample size, there's going to be some amount of LD that's still too much and some amount that you've already...
I get that. It would be very interesting to see what that looks like across the European population, but also, it's not the case that the functional variants are just distributedly randomly with respect to the LD. Right. So there's also the just practical how are the variants distributed with respect to LD.
Yes. And I'd like to make the point that given the choice between doubling the sample size of UK Biobank or having a second UK Biobank in another population, because of this interaction between sample size and LD.
Always get the other one.
No, exactly.
That one I got.
Even at like much lower sample size for fine mapping specifically, because the LD is so important.
And then the kind of equivalent, I totally buy that, the equivalent question to Anshul, but now not thinking about genetic samples, but thinking about functional, other ChIP-seq data. How far away are we from saturation in some sense?
Yeah, so I used to think we were pretty close. And then the example I showed of that enhancer, it's right next to HNF4A actually. I showed it in many different tissues, and they essentially have highly overlapping peaks, right? We usually have defined coverage as basically number of bases already covered by some element. At the sequence level, they seem to be regulated very, very differently. And if we hadn't got all that information in all those different cell types, in fact, some of them are colon-like cell lines versus primary colon tissue, and there are subtle differences in cofactors. Now, of course, all of this needs to be validated, but there's strong evidence from footprints and ChIP-seq and so forth. So the definition of coverage is sort of depends on what you're interested in. If you're simply interested in, did I cover the element in some cell type or tissue? Sure. If you're trying to figure out what precisely is driving that element in terms of sequence, there seems to be very subtle regulation within elements. So I think that changes the ballgame a little bit. I think you need to get the cell type exactly right and the cell state exactly right to figure some of those aspects out.
Okay, you didn't really answer the question. That's fine.
Really impressive talks, really interesting. So my question is for Anshul as well. So you did mention that for those out of distribution cases that you mentioned, for example, the role of ancestry as a future direction to follow. So I was wondering if you have any preliminary results or prior at least of how would that may be affecting sequence variation in these network relationships. So how would that can that be integrated?
Yeah, so that's a really good question. So all the current machine learning models that I've seen so far have trained on the reference genome. I think that's a reasonable first approach, but what they explicitly do is mask out the effect of variation that they do not see in the training data. So if you take you take a sample that has a specific configuration of variants and you map it, you have functional data in that same sample. You learn a predictive model, but you use the reference genome instead of the actual sequence, then the model is explicitly learning to mask over the effects of variation. And then those same models are being used to predict the effects of variation. That's the reason they really don't work. And it's actually quite easy to switch the input representation instead of using like regular ACGT, which usually you show as 4 channels in the input. You can make it 8 channels so you can get deployed. So you can say like ACGT maternal, ACGT paternal, or you can have ACGT and instead of having it as a binary encoding, you can add allele frequencies or you can add probability of each base. So it's very easy to transform the models to account for personal variation. But that actually tells us that we need information where the genetics is linked to the molecular phenotypes. Somewhat similar to what we have for EQTLs, but we don't have that kind of data for chromatin phenotypes. And I think chromatin phenotypes, in my view, are actually more powerful than expression phenotypes for looking at proximal molecular effects of variants.
That's really interesting. It would be great to talk more offline. Thank you.
Judy?
We probably don't have enough data in non-Europeans to answer this comprehensively, but it seems like one goal of ICDA might be to map the prevalence of diseases in different populations with polygenic risk scores, which we know don't go across populations well. And then given that background variation, if you have a causal allele, you're going to see very different effect sizes. It just seems like it's kind of empiric activity that needs to be done by ICDA. So any comments or thoughts?
Sorry, you're saying start by mapping prevalence of disease in multiple populations and then use that to help calibrate the polygenic risk scores?
Well, oh, no, not to calibrate but like what's the relationship between polygenic risk scores and population prevalence of diseases? So like going back to the point Kari made, you want to study it in the at-risk population, right? So it just seems like there should be a comprehensive listing of this somewhere.
Yeah. I can comment maybe. Yeah, I think it's a really important point. I think we know well that the PRS is done are not completely portable across populations. And so, this is obviously justifying discovery efforts in different populations, but perhaps we can also think about how we can leverage the information that we acquire in a population to accelerate that effort. And it's not only the point that Nick was making this morning about accelerating knowledge, but also accelerating, having biological priors for where we look for these loci. I think that would be very, very interesting to at least to begin to test. I guess the other interesting thing is that we know beyond the ethnicities, also environments change dramatically between different populations and
right.
Exposures and etc. And so I think it'd be really nice to again, even just looking at portability as a function of local environment. So it'd be super exciting, I think.
And just to add on to that, a parallel question we had to address in the imputation panel days was if you have a specific data set you're trying to impute into and you have a global reference panel, are you better off using that entire global reference panel or just the ancestry matched part of it? Yeah. And essentially the bottom line was in most cases you won't hurt yourself much by using the entire panel. Yeah. And we need to now ask the same questions with polygenic risk scores, and I think the jury is still out whether you're better off using a meta-analysis from a larger sample size, more powered population, but it's not a strict ancestry match versus a smaller ancestry matched one, or perhaps some combination of both that we need to come up with. Yeah. Manoj.
So it may be a little bit sort of off the main track, but I think it's very relevant to, especially from Nicole's presentation. So one of the problems of putting together, let's say, networks of causality, so from variant to one phenotype to the next one and so on, is that, if we assume that the technical variance and overall the unexplained or the unknown variance is about the same, then and it's not too much, then you kind of have a little bit of control. The problem is that none of these assays have exactly the same variance. Yeah. Proteomics has a different variance, RNA-seq has another one, ChIP-seq is a different, genotyping is different, and what we've seen is that when you try to put together in causal models assays that have very different variants, technical variants that you cannot even measure or estimate, you have a big problem assigning causality because the model goes all over the place. Usually it just shows a large amount of independence when in fact it's not. It's just that there's so much variance that completely distorts the causality that you should be detecting. Mm-hmm. Given that we are actually going into cutting edge technologies that we're less and less knowing that variance, and we have less control of this because we are actually touching that edge. How are we going to deal with causal modeling? I don't think it's an easy problem to deal with.
Yeah, it's super difficult. I agree. I fully agree. And I fully agree with your statement. I guess the one thing that we can consider is that, if you look at models such as some of the ones that I showed today, and, the more dimensional the data, the better grasp we have of unmeasured, unknown technical confounding, right? And so these methods where you have a factorial representation of the data is able to model some of the technical variants. Yeah, and I think there's a lot of work still to be done in understanding how to do this properly and how robust that is, but at least the frameworks exist. And so now the question is, and so you could apply it in different ways. You could have, even this gene environment modeling, where the modeling is done on the technical variants to understand better about what are the things that we should be worried about. It's a concern for transQTL effects as well, right? You might be removing the effects with once you remove the technical variants. But I think, part of that is, can we leverage this type of, slightly agnostic frameworks to learn more about what is the things that we should be worried about, and particularly for false negatives, causality that you can't see. The other point I wanted to make is also, I think we're going to move away from a very binary definition of what is causal and what is not, and really begin to understand that there are dimensional causality that could be environment dependent, context dependent, and so this ability to define what is a proportional causality that, rather than an absolute answer, is perhaps going to take us in a slightly better direction.
So we should probably start to wrap up. Terry, you want to ask your question, and then we'll decide if Ewan gets a second one or not.
But just very quickly, wanted to thank Nicole for doing that analysis. I'm sorry, Hilary, for doing that analysis. Thank you. That was very cool. It may well be the best case, obviously, because hemoglobinopathies are so common in those parts of the world. But it would be really neat to generate those data, and they can be returned as a return data set to UK Biobank, as well as posted on the NEAL server and the Riva server. So I'd encourage you to do that.
Great. We're definitely planning to do that for the summary statistics, and are wondering about the logistics of the LD.
Does anyone want to hear Ewan's next question?
Sure.
Anyone?
Let's hear his talk.
Is it quick?
Yeah.
All right, come on.
No, because it's very different. Let me just briefly say that just a meta thing. I'm channeling my inner Carlos Bustamante and Peter Donnelly here, both of which I think would say that I think we have to move from a world where we think about humans as coming from these separated populations, and we treat admixtured individuals as just these kind of complicated people that we exclude from analyses. And we have to turn it around and think about everybody being complex ancestries, of which some people are smoother complex ancestries, in which we do discovery. And I think something that I learned from Peter and his work from Genomics PLC has been that the fact that we need these rather homogeneous subsets for discovery of genetics does not mean we need to apply them to homogeneous people in terms of their ancestry. I think we need to really, really make sure we think about both the mapping problem, the discovery problem, but also the application problem in a kind of complex ancestry. Make the default that everybody has complex ancestry, of which some people are going to be easier to solve.
Nicole, can you be pithy, or Hilary?
Yeah, so I 100% agree, and we're thinking now about statistical methods for how to get beyond this, and I want to highlight that the reference panel paradigm does rest on this idea of distinct homogeneous populations, and so I think we definitely need to develop the models and then get the data to fit them.
Yeah, we need to leave that behind, though. I want to have a one population model.
Okay. Let's thank our speaker panel. You guys are awesome.